So good afternoon all. I'm Michelle Richards, the Director of Building Operations here at TRI, and thank you for joining us for TRI's first Biotech stage event. This series is an opportunity for industry that's based at TRI to let you know about their areas of research, their development and their clinical interventions. These events aim to provide an opportunity to hear the latest biomedical advances from our newest tenants at TRI, to connect to them and inspire research, clinician and industry interfaces. Today's seminar will focus on the brain, heart, eyes or stroke, heart attack and retinal disease, each of which have a huge impact in our communities. We're going to have 3 speakers today and then a panel session at the end. So I'll ask everyone to save up their questions for that panel session at the end. I'd now like to invite Brenda Castro to the stage. Brenda is a pharmacist and obtained her master's and PhD degree in pharmaceutical sciences at the Federal University of Minas Gerais in Brazil. Brenda is passionate about discovering and improving therapies for eye diseases and has been working with nanoparticles, polymeric implants, peptides and gene therapy for different ocular diseases, especially uveitis. She was awarded a scholarship to spend a year of her PhD at the University of California, Irvine, where she worked with viral and non-viral nanoparticles for ocular gene therapy. After completing her PhD, she moved to Australia in 2021 and started working with the Latent Vision Foundation, Ocugene, as a postdoctoral scientist investigating novel AAV-based gene therapies for eye disorders. Thank you, Brenda.
So good afternoon, everybody. So I'm Brenda Castro. I'm a postdoc scientist at the Latent Vision Foundation and its startup, Ocugene. So here we develop next generation retinal therapeutics and gene delivery vectors. And we have 2 main platforms. One is the discovery of next generation customized AAV vectors for optimized retinal transduction. So we used the directed evolution technology to screen random peptides libraries using human eye cups as a model. To select vectors with efficient transduction in retinal cells that would induce low immune response and also to be suitable for intravitreal administration. So a very common route of delivery in gene therapy for eye disease is the subretinal, but the intravitreal has many advantages. It's easier for patients and clinicians, has a low cost, and that's the one that we are focused on. It's also part of this platform, the establishment of a manufacturing process for the production of GLP and GMP grade vectors. Our second platform is the development of controllable gene expression systems for precise, safe, and long-term effective therapy. So we designed AAV constructs containing a inducible gene expression system to control the expression of transgenes with non-therapeutic effects in eye disorders. So these systems, they are developed for turning the gene expression on and off, or to modulate the level of expression in a quantitative and temporal way. And so to turn the system on and off or to modulate, we use inductors that can be easily administered by the own patient using oral or topical delivery routes. So we are a very small company and we are in very early stage. So we have now 2 vectors. We call them C1M1, C1M2 for the optimized retinal transduction. And we have 4 constructs. So we work with the tetracycline inducible system and the QMate inducible system to control the expression of genes with anti-inflammatory or anti-angiogenic effects. I'll talk a little bit about our first platform, these novel AAV capsids. So having in mind that viral tropism is determined by the sequence and the structure of capsid proteins that interact with cell receptors. And our previous work showed that neutralizing antibodies against AAV8 is less frequently seen in human vitreous. So we chose to work with the recombinant AAV2/8, which is rep genes from AAV2 and cap genes from AAV8, in order to find a variant with more efficient transduction. So we used the ICUPs as a model. So we screened this vast library of peptides in human ICUPs, and we select those which would highly transduce retinal cells, especially those cells in the outer retina, which is a layer of part of the retina highly affected in the most common disease. And since we want to use intravitreal injections, so we have to have in mind that these viruses will go through from vitreous through the retina to reach this part that's like deeper part of the retina. So we select the variants, these recombinant AAV2/8 with triple mutations and also the P5 promoters. So this is the AAV8 capsid. So these variants that we chose, they have this peptide inserted in the loop 4. So this is a loop 4. Sorry, I'm showing here. So we have the loop 4 over there, and in the loop 4, we inserted this tiny peptide in a region that affects the interaction with cells, and we aimed at getting better tropism in retinas. So this is just a little bit of our data. So in vitro studies using immortalized retinal cells show that our capsids, our novel viruses, they did induce retinal cells better than the parental viruses, which is a 2/8. And with the results similar, comparable to the 7M8 capsid. So 7M8 is a AAV capsid widely used in ocular gene therapy, and that's been tested in clinical trials. So that's like our reference. So we have results comparable to the 7M8 and better than the parental virus. And using ex vivo model, like using ICUPs, human ICUPs, and the luciferase as a reporter, we confirmed that our capsids, they transduce retinal cells. And when we compare over time, we see an even better performance compared to 7M8 capsid. And so, and this was done in like retina explant and another explant containing the RPE cells, choroid, and sclera. So this is just like a brief, some brief results, just showing that our capsids indeed is doing what it's supposed to do. About our second platform, so we are developing these controlled expression systems. We work with the tetracycline and the QMate inducible system. They work similarly. So in the absence of the inductor, gene expression is prevented by the repressor. And when the inductor is added, it binds to the repressor and then gene expression occurs normally. These inductors, they can be given, I mean, we aim at giving them using oral and topical administration routes. The inductor for the tetracycline inducible system is doxycycline, that's already used and safe for oral delivery. And for the QMate system, the inductor is cumic acid that we are formulating to be delivered using eye drops. So we designed these constructs and using different reporters and transgene and therapeutic genes. And some brief data shows that we, using mCherry as a reporter, we see that like cells transduced with the QMate system and treated with the inductor, cumic acid, that they do show some fluorescent cells comparable, like similar to CMV expression, like a constitutive expression, and the cells transduced and not treated with the With the inductor, they didn't show much fluorescence. Using luciferase as a reporter, we see that we turn the system on. When we turn the system on, we have higher expression. When we turn it off, when we remove the inductor, it slowly goes back to baseline values. And then a month later, we turned the system again on. So we treated the cells again with the inductor, and we again saw increase in the expression. But for the QMate system, we see some leakage in the off system, the system not treated with the inducibles, the inductor, that we are working on this. For the tetracycline, we actually saw a better result. So we can, when we treat the cells transduced with the AAVs containing the tetracycline inducible system encoding luciferase, we see a peak in the expression, and when we remove, it slowly goes down to baseline values. And a month later, when we treat the cells again with doxycycline, we again saw this high expression, and the off system, the untreated cells had insignificant production of luciferase, and our expression was very close, was significantly similar to CMV expression. So we focus on two pathological processes at Ocugene. So our therapies focus on neovascularization and inflammation. And it's important to target this process because they are common in many ocular disease. So inflammation, for example, it's a process involved in the pathogenesis of uveitis, dry eye, glaucoma, diabetic retinopathy, age-related macular degeneration, while neovascularization is also present in corneal neovascularization, tumors, diabetic retinopathy, age-related macular degeneration. So by targeting these processes, we expand our products to be used in different ocular disease. So among these diseases, we chose age-related macular degeneration, AMD, and uveitis. They are highly prevalent disease, and the current treatment is a burden for patients and clinicians. It's associated with failure and adverse events. So age-related macular degeneration is the leading cause of blindness in the developed world, and it's increasing, the prevalence is increasing with the aging of the population. So it's estimated to be affecting 288 million people by 2040. Our products, they are aimed, we are targeting wet AMD, that's the neovascular form of AMD that's characterized by this abnormal growth of choroidal vessels into the retina that evolves to leakage and rupture of these vessels that results in exudation and severe vision deterioration when not treated. The current treatment is with anti-VEGF agents that are effective, but it requires frequent intravitreal injections, like every 4 to 12 weeks, which is very, which is uncomfortable for patients, has a high cost, many complications associated, and ends up having a low compliance. And if the patients don't follow the treatment, they lost their sight. And so there is a need for long-lasting therapies with improved safety and efficacy for AMD, and this can be achieved with AAV-based gene therapies. Our proposal for this one is this construct containing endostatin and angiostatin. So they are endogenous inhibitors of physiologic neovascularization. So it's a bifunctional protein that we're going to package into optimized vectors, the vectors that we have developed, and add the controlled expression system to control and modulate the expression, turn it on and off when we want, when it's necessary, depending on the disease course, and to be delivered in intravitreal injections. That's easy for patients and clinicians. Our other target disease is the uveitis. So uveitis is the inflammation of the uveal tract and adjacent structures, it accounts for 10 to 15% of all cases of blindness in developed countries. And it's even worse in developing countries. About 35% of all patients with uveitis, they have blindness or visual impairment in at least one eye. And this is a big problem because different from AMD, uveitis affect mostly young adults of working age. So these represent an important economic and social impact because these people cannot work anymore and then have like depression. It's not independent anymore. So our therapies focus on posterior non-infectious uveitis. So posterior is just the location of the inflammation, which is in the back of the eye, which is even harder to treat with the conventional therapy. So current treatment for uveitis is with topical and systemic steroids. When you think about posterior uveitis, to have these steroids to reach at therapeutic concentrations, the back of the eye is a big challenge. So sometimes it requires multiple instillations, systemic administration, high doses, and which is associated with important and severe adverse events. And when the inflammation is not controlled with steroids, other agents are introduced in combination or not, like alkylating agents, immunosuppressive drugs, biologics, and they all have costs associated, adverse events, so it's not really good for patients. So there is this unmet need of more effective, safe, and specific therapies for uveitis, and which can also be achieved using AAV gene therapies. So our proposal for uveitis is this construct containing the anti-inflammatory cytokine, IL-10, and an antagonist of IL-1 to reestablish the homeostasis. So these construct will be packaged in optimized vectors. We're gonna add the controlled expression systems. It's a dual active system, so we're acting different parts of the inflammatory process, and to be also delivered in intravitreal injections. So this is just a little bit of our work. We are, as I told you, we're a small company. We're doing at the moment like basic research, and we are working on developing ex vivo models, disease models, because we have easy access, relatively easy access to eye cups. So, and it's a great model like to establish before moving towards preclinical studies with animals. We are also working on optimizing our AAV production, thinking about having GMP grade vectors for the future. Yeah, thank you all so much for your attention.
Thanks, Brenda. Again, save your questions up until the panel at the end. I'd now like to introduce Stuart Newman. Stuart is the CEO at Therapeutics Innovation Australia. And since completing his PhD in Antarctic biology from the University of Tasmania, Stuart's built up a considerable experience of science policy, pharmaceutical R&D, grant funding, IP management, business development and commercialisation in the university and not-for-profit sectors. Stuart joined TIA as CEO in 2017, and under his leadership, TIA has focused investment on the gap between research and development of high-value therapeutics, including pharmaceuticals, biologics, vaccine, and cell and gene therapies. He's also devised an innovative infrastructure access voucher scheme, and he's a graduate of the Australian Institute of Company Directors. Thanks, Stuart.
Thank you, Michelle. Thanks, everybody, for this opportunity to speak with you today. TIA has been a tenant of TRI for about 11 months and 1 week. And we're delighted to announce we're going to re-sign to stay another at least 12 months, we're finding it to be an excellent place to locate ourselves. It gives us access to our valued stakeholders. Now, this is going to be a bit of a change of pace because we're not a company like Infenza and Ocugene in the sense of developing therapeutics. We are a not-for-profit company that supports companies developing therapeutics and also researchers in the academic and SME space. So terrific. So I'm just going to go through a little bit of an introduction of our mechanisms of how we support such research and talk about our voucher scheme, which opened yesterday, and encourage people to take a look into that. So we are part of one of the best kept secrets in Australian science, which is the NCRIS scheme. It's a network of, might have been me 25 by now, research infrastructure projects. It's all about making research infrastructure on a number of scales accessible to industry and academic researchers alike. It supports all the research that Australia is, or would like to think it is, world-class in, and that includes the medical product space. We are part of a bigger picture. In the NCRIS program, there are other projects that we have direct links with. For example, Bioplatforms Australia is the omics powerhouse in the NCRIS network. The National Imaging Facility, with whom we share an office on the 7th floor of this building, focuses on preclinical and clinical imaging technologies. Phenomics Australia is in the in vitro and in vivo, ex vivo I should say, disease modelling space. Population Health Research Network is in health-linked data, and as ourselves, of course, we have some joint appointments and joint initiatives, and we often refer projects to and from each other with reckless abandon, just to make sure that researchers have access to the right facility at the right stage of their project. But back to us, we enable translation via our consortium. We don't have labs or facilities ourselves as a company. We invest in the capacity and capability of facilities such that researchers can approach these facilities and gain access to their expertise at reasonable financial rates. We also have a additional voucher scheme which incentivizes collaboration, which I'll talk a little bit more about later. We have a number of staff around the country, Melbourne, Sydney and Brisbane, who act as brokers or business development kind of roles to go and find projects that wherever they may be, either via knocking on doors or going to conferences, and they direct them to the right facility. So where we place ourselves, perhaps rather foolishly, is right in the valley of death, right in the gap between the discovery and the point at which projects start to become a bit more economically viable and attractive. That's pretty much where we put ourselves. And our three broad capability areas are arrayed around product types. First, we have a small molecule pharmaceutical group. It's a fairly nice integrated pipeline from compound management through to in vivo preclinical models. We have a biologics and vaccines capacity in University of Queensland, CSIRO and University of Technology Sydney, and cell and gene therapy network around the country as well. I won't go through all of these because there's quite a few. We have a reasonably wide national footprint in all these areas. I'll start with very briefly on the National Biologics Facility. It's a fairly comprehensive, available set of technological capabilities, all the way through from discovery support, mRNA and other kind of RNA production at the lab scale and lab quantity, through to big effort in mammalian cell line development, mammalian expression systems, beginning of QC assay and process development for a biological product and the vaccines. And finally, opening last week, actually, clinically enabling manufacturing and training capacity. That's up to 200 litres. I should say CGMP is on the way, but not quite yet achieved. We also offer training in this environment. So we have a group of small molecule development facilities, starting with compound libraries available through Compounds Australia, a network of high throughput screening facilities around the country, some hit to lead medicinal chemistry located at Monash University. Also at Monash, we have offer DMPK, guided lead optimization, and some preclinical efficacy models at UQ. And finally, I'm whizzing around these quite quickly, but details, just come and ask me if you need any more details. Have a network of 5 facilities for development and manufacture of cell and gene therapies. In Brisbane, it's a Ocugene facility. 2 in Sydney, including development in AAVs. Peter MacCallum Cancer Institute in Melbourne, and Royal Perth Hospital in, spoiler alert, Perth. And a couple of nice case studies that our facilities have been involved with. Kira, local Kira Biotech has raised $20 million worth of Series A funding based on a lot of work that was carried out with the assistance of our local biologics facility. Unfortunately, this didn't quite save the world, but the CEPI-funded molecular clamp vaccine developed at the University of Queensland was advanced from DNA to a phase 1 trial in 6 months, showing that we can do it. We can do it right here, and a vast proportion of that work took place at the TA's facility at University of Queensland. One of our first voucher schemes, voucher awards back in 2017, was to help to generate a T-cell bank, which is now being used in phase 1 clinical trials at Berghofer. I just want to put this placeholder here because we are developing an RNA products capability, but can't say too much about that at the moment, but it's very exciting, I should say. Now, how to find us? There's a few ways. The best way is to come and knock on our door on the 7th floor, but if you can't, by all means, contact us via our email, or we have on our website what we call our Pipeline Navigator, which is a searchable database of facilities, not just our facilities, but accessible facilities around the country. In terms of accessing TIA source funded capabilities, you can contact us and we'll point you in the right direction. You can directly approach the facilities as well. They mostly operate a tiered funding model for research users, SMEs, and also pharma and biotech. But what I would particularly like to talk about, which scheme we launched yesterday, is our Pipeline Accelerator scheme. This is an additional subsidy for projects. Since we started the program, we've made 115 awards with nearly $4 million. It's essentially a voucher system up to $50,000 to defray further the cost of accessing one of our facilities. So equally open and welcome to SME researchers or academic researchers or tech transfer offices or hospitals or what have you. International applicants are also welcome. We do this every 6 months and there's a pretty rapid response time. So happy to answer more questions about that or talk about that more offline. And we try and make awards by mid-December so people have a very Merry Christmas. Key features of this scheme, we don't seek any IP. We are enablers, not investors in that sense. We want to reduce the cost of access. We don't want to pay for it all. it's a subsidy, not a grant. There must be some matching funding involved in some way. And yes, so I would just like to thank everybody for their time and invite any questions during the panel session, and come and see us, knock on our door on the 7th floor. We're in the same office as Mark. Thank you very much.